Okay, it appears we are live, so as usual, I'm just going to make sure that everything looks good on my end, on YouTube, and then we will get started. Hi, everybody. Thank you for coming back for part 2, those of you that are here. Let's see. Do we have sound on YouTube? Yes, we do. We're good. Okay, cool. So let's see. Just move this window out of the way. All right. I see people are here. Hello, everybody. Delightful. So we are here with part 2 of my review of Replacing Darwin by Dr. Nathaniel Jeanson, currently affiliated with AIG. I can get rid of this. I know the sound works now. Get rid of that. Okay, so let me start with a little recap. This is part 2 of the review of Replacing Darwin, this monstrosity right here. Last night we covered actually most of the book. I skipped the early chapters because the early chapters are really more or less non-offensive overviews of genetics, and they're more or less okay. There's nothing so objectionable I feel the need to point it out here. But we covered in some detail chapters 3 through 7 last night. Now, we didn't cover these chapters in a ton of scientific detail. This is a book review, not an analysis of Jeanson's scientific arguments. I've done that elsewhere, and you could find links to many of those videos in the description. You can also find a link to Evograd's wonderful series on Replacing Darwin. That link is also down in the description, and he goes into a ton of detail in terms of actually examining Jeanson's scientific-sounding arguments. Now, I am not going to do that here, one, because time constraints, and two, is that having read this book very carefully, I think it's clear that Jeanson is not trying to make a scientific argument aimed at a scientific audience. So I think treating it seriously in that regard is not the optimal way to approach this work. This is a work that is based on the presupposition that a literal reading of Genesis is correct, and then we're going to torture the data until it confesses that that literal reading is correct. And that argument is aimed not at a scientific argument. It's not aimed at, like, I'm an evolutionary biologist. It's not aimed at me. It's aimed at a non-scientific, already creationist argument. It's not trying to persuade. It's trying to reassure and provide things for that audience to say. So what we're going to do tonight is we're going to finish this up. We are going to do chapters 8, 9, and 10. I literally only have a couple of things. I think chapter 9, there's not too much to talk about, but there's an interesting sleight of hand that goes on in there. And yeah, we're going to go through it. And you're going to see some pretty egregious examples of the categories of things I talked about a lot last night. So to recap, there are kind of 3 things that I want to point out that Jeanson does here. So one is he bases his arguments on assumptions or assertions about how, like, evolutionary processes or genetics work, and these assumptions often are untethered to reality, let's say. The second thing that he does is he egregiously strawmans the evolutionary position, and I provided some examples of that yesterday, like how what his explanation for the bad design argument and why that is considered things that are bad design, like the recurrent laryngeal nerve in giraffes, for example, why that is considered evidence for evolution. And he just misrepresents things. He presents straw man arguments rather than the actual arguments. And then the third thing that he does, and we're going to see more of this here, is he just doesn't seem to want to operate within how science works. And specifically what he loves to do is make predictions and then say, because I've made the predictions and the predictions have not yet been tested, therefore the hypothesis underlying those predictions has not yet been refuted. Therefore, you have to take it seriously and put it on equal footing with the alternative, which is to say evolutionary theory. And until you test my predictions, you can't say I'm wrong. Therefore, you have to say we don't know. We have to consider my explanation just as plausible. And that is a ridiculous representation of how science works and how scientific discourse and arriving at answers using science works. Jeanson and other creationists that lean very hard on Jeanson are very fond of saying that the gold standard of science is making predictions, is making testable predictions. That's the gold standard. No, the gold standard is making correct predictions. The gold standard is making testable predictions, testing them, and verifying that they're correct. Jeanson starts at make predictions, and then he stops at make predictions. He doesn't do the rest of that. And that's something that we see throughout this book, just basically saying, here's a prediction, therefore you have to take me seriously, with no underlying support to show that that prediction is actually correct. Okay, so let's get right into it. We are going to jump into chapter 8 here. We're going to cover chapters 8, 9, and 10. And just like last night, I'm going to go through the pages in order within these chapters. And I am going to just show you the examples of things where he's just making bad arguments, where he's setting up straw men, and where he is kind of committing crimes against the scientific method. Okay, so that's what we're going to do here. So let me, if you were here last night, you remember that we've got 87 of these screenshots here from the book, where not all of them, so 3 of them are the intro stuff. But then what I've done is just take pages where there's interesting things, and I've circled and will point out the interesting things where there's a problem with what Jeanson is saying. This is by no means an exhaustive list. I actually have about twice this many, but in preparing for this, I cut it down to the 85 or so that I thought were the most egregious and worth looking at. So let's we're going to get all the way down here to 801. So it's by the way, just the notation here. 08 refers to the chapter, and then 01 is the number screenshot for me in that chapter. This is obviously the ebook version that I'm working on here, so your page numbers are going to be different from mine if you want to follow along, but you can always just go by the chapter and then kind of control F, whatever sentence it is that we're talking about. So let's get right into it. Let's get right into finding Jeanson's bad arguments. So this is in chapter 8. He just right off the bat starts with something that is wrong. He says, consequently, evolutionists would be justifying and claiming that applying the label functional to 80% of the human genome is premature. That's actually correct. They would also be justified in withholding judgment about the function of most nuclear DNA sequences. Correct. Follow me to the next paragraph. But in practice, evolutionists have often swung the pendulum in the other extreme. So what he's doing here is similar to what he did with the argument about constant mutation accumulation rates, where he said, evolutionists, you say things like radiometric decay, the speed of light, all that stuff happens at a constant rate. So to be consistent, you also need to say that mutation accumulation happens at a constant rate. And what he's saying here is like, sure, you can say we don't know what everything is that's functional in the human genome, but you really tend to err on the side of saying stuff is functional, and to be consistent, we should go in that direction. Now, he goes on to make an argument here about the similarity between human and chimp genomes and shared mistakes. And he makes an analogy using exams. If you have 2 students that have exams and you think one's cheating off the other, and what you can do is you can check the wrong answers and see if the wrong answers match. Because if the wrong answers match, then that's strong evidence that they're cheating. And as someone who has lots of students every year and has actually done that, that's true. That is true. If you find a bunch of wrong answers that are like identical, that's pretty strong evidence that one of them is copying off the other. Or in this analogy, that they share a common ancestry. What he kind of misses here, right? If you look right here, He says, similarly, the evolution have argued that the existence of shared genetic mistakes between humans and the great apes indicates that humans plagiarized, inherited, other nuclear DNA from a common ancestor. Yes, but you can also look at the functional parts, and you can see how, like, humans and chimps tend to be more similar than humans and gorillas, or chimps and gorillas. And those three tend to be more similar to each other than any of them are to orangutans. Now, there are exceptions to that due to things like incomplete lineage sorting and occasionally horizontal gene transfer, but for the most part, those patterns hold up. And the interesting thing about this is that Jeanson is generally comfortable with that kind of thing within families, not for apes for obvious reasons that we'll talk about that later with humans, but in general, he says this is nested hierarchies within families show the common ancestry. The problem is you can do that same kind of thing looking at shared mistakes. There's no reason to stop at families. You can just keep on going up all the way up to, phylum, kingdom, domain, and you find the same thing. So the argument that Jeanson is using to say, hey, common ancestry looks that way and we generally can accept that, you can just keep going. And there's no barrier where that argument stops working. So now we're still in chapter 8 here. Again, the results are only biochemical experiments, not genetic knockouts, but they set a trajectory that points toward pervasive genome-wide nuclear DNA function. Now, this is, of course, talking about ENCODE. So ENCODE documenting biochemical activity among wide swaths of the human genome. About 80% of it, give or take, in their initial major publication in 2012. Now, it's worth noting a couple of things here. One, they've revised those numbers, and the biochemical activity that they identify is not a good indicator of function. I have a whole video on this. It's linked down below. It's the ENCODE one. The important thing, though, is that ENCODE actually kind of walked back their numbers, and they're in their later publications, like their 2014 paper, they're a little more careful about their language. And they describe a genome that has maybe more function than we expected. They probably overestimate it still, but they're saying if you look at things like transcription and levels of transcription, it's 30, 40, 50, maybe 60%, whereas previously they were saying above 80%. And they acknowledge that all that transcription, a lot of that's probably not actually functional. It's spurious. So the second thing I want to say about this, in addition to ENCODE not buying what Jeanson is writing here, even though he gets it from them, the second thing is that creationists tend to treat junk DNA as an unknown. They tend to use junk DNA as a stand-in for stuff we don't know what it does. That's not the case. The stuff that we call junk DNA, and I know some people don't like that phrase. I don't care. I like that phrase because there's DNA. It doesn't do anything. It doesn't hurt you, but it doesn't have a function. It's junk DNA. And the reason we can call it that is because we know what it is. It's well characterized. We know where it comes from. We know what activity it does, and we know that it doesn't have a selected function. Therefore, it's junk. We know what it is. Junk is not a placeholder for we don't know. It's a specific descriptor for stuff that we've characterized. And creationists often miss that and present it as we don't know. Well, we're going to find out in the future. No, we figured it out for a lot of the genome. In fairness, there's like 15, 20% that's not well characterized. I don't know what we're going to figure out for that part. But for the rest of the genome, we get a good handle on this. We're pretty good. We know it's there. Okay, so now let's continue on. This hypothesis leads to expectations about function at the site where the proposed. Oh, we're talking about chromosome 2 fusion here. Here we go. He's saying it's not a fusion. It's not a fusion. Because that's the creationist position. You see Jeffrey Tompkins is the big one that writes on this, and his stuff is just awful. And we're going to get into it a little bit here. At the site of the supposed fusion in human chromosome 2, evolutionists expect to find a genetic scar. I guess what he means by that is a head-to-head degraded telomere. Okay. They have even claimed that such a scar exists. It does. There's a head-to-head, it's a telomere, a degraded telomere, and then it flips, and there's a telomere at the opposite orientation right next to it. So you've got a head-to-head telomeres in opposite orientations that are degraded. That's what's at the fusion site. In contrast, recent investigations have demonstrated that the supposed fusion site does not bear the scar of an accidental chromosomal crash. That's just not true. Rather, the site sits in the middle of a functional gene. It's a pseudogene. It's a transcribed pseudogene. There's a bunch of copies of this pseudogene in the genome. They are transcribed. Whether they are functional is up for debate. I don't believe they are translated. but it's questionable whether the RNAs have a function. So it's not a functional gene in the traditional sense, in that it does not code for a protein. I'm like 90% sure of that. And the purported fusion sequence appears to participate in regulation of this gene. Okay, so here's the thing with this. This gene, actually, I'm going to look this up. Indulge me for a second. I apologize. I'm going to look this up because I forget the name of the pseudogene. I'm going to say chromosome 2 fusion pseudogene. As soon as I see it, I'm going to know it's like the yeah, chromosome 2 fusion gene. Sorry, everybody. I'm forgetting what the name of the pseudogene is. If anyone in the chat remembers, it doesn't matter. But anyway, it's a pseudogene where there's a bunch of the pseudogenes in this family within the genome. The interesting thing is the one on chromosome 2 is in the middle of the chromosome. It's at that telomere head-to-head fusion site. Thank you, Walker. It's, yes, the DDX11L2 pseudogene. Thank you. That DDX11L family, I think, is the family. I forget where you draw the line at where the family, or DDX11 family. There are a lot of those. You know where they all are throughout the rest of the genome? Every single other copy of the pseudogenes in that family? They're all in subtelomere regions. They're immediately adjacent to telomeres. The one in the middle of human chromosome 2 is the only one that is not immediately adjacent to the end of the chromosome, right? That's it. So that's one. That functional gene, it's a pseudogene, not a true functional gene. Those genes, there's a bunch of pseudogenes in that family. Every one of them is next to a telomere, except for the one in chromosome 2. It's the one in chromosome 2 is next to the degraded telomere. The last thing I'll say on this is that in terms of being expressed across that gap, it has an alternate transcription stop site. So some of the transcripts stop before the telomere fusion site. Occasionally, it'll read through that, and there's one more part on the far side. And occasionally, it will transcribe across that gap. It doesn't happen all the time. It doesn't even happen most of the time. It happens some of the time, it'll transcribe across the gap because you have an alternate transcription stop site on the far side, and the one on the near side is leaky, so it doesn't stop 100% of transcription events. That's what we're talking about here. It's not strong evidence of functionality. It's not showing that that site is part of a functional thing. If it was part of a fully functional gene, the site itself would be in an intron. It wouldn't actually be part of the coding sequence of the gene, so it wouldn't participate in the protein sequence anyway. So that's what's going on with human chromosome 2 there. Eventually, I'm going to have to do a creation bits video on human chromosome 2. Just haven't gotten to it yet. All right, here we are. Just like for mitochondrial DNA, the fact of nuclear DNA mutation suggests that nuclear DNA differences work like a clock. Fine. Again, to be consistent with the evolutionary practices in geology and astronomy, we should assume that the nuclear DNA clock ticks at a constant rate. He's doing two things here. He's doing a bait and switch, which I don't think I pointed out last time because he's I did point out last night that he's using units incorrectly here. He's saying mutation rate when he means mutation accumulation rate. But that's what he means when he says mutation rate. He means the rate of accumulation. And then he's doing the misrepresenting that it should be constant, which no, we know there are factors that are going to modulate that rate. Things like selection and drift and inbreeding, right? So there's wrong there, but there's also this ridiculous logic that he's saying evolutionists have to follow. Well, you say geology, astronomy, physics, you see all that stuff is constant. So therefore mutation rates have to be constant too, which, like, what? I should say mutation accumulation rates, because that's what he actually means. In a way, he's accidentally correct when he says something like this, because mutation rates, the rate at which mutations occur, those are actually pretty consistent, right? So like in humans, we have DNA repair mechanisms, our polymerases make an error at a predictable rate, yada, yada, yada. The rate of mutation is pretty constant. But what you see variation in over time is the persistence of those mutations due to things like the germline bottleneck, selection, drift, inbreeding, that kind of stuff. And Jeanson, despite using the words for the former, is talking about the latter. He's talking about accumulation. And that's the problem here. So now we're going to talk about time. Okay, this is one of my favorites here. This is the big straw man when it comes to time dependence, because it shows that Jeanson either completely does not know what, like when I say things like selection, drift, and inbreeding decrease the number of mutations that persist, this passage shows that when like Jeanson hears something like that, he either doesn't know what it means or he's just misrepresenting the biochemistry, the genetics, the basic biology underlying this stuff to give a talking point to his creationist audience. I don't know what the explanation is, but let's read this paragraph. This is a big one. So consider another potential evolutionary explanation. In the realm of mitochondrial DNA, evolutionists have already discussed a phenomenon called time dependency. In other words, evolutionists have suggested that the mitochondrial DNA clock ticks at different rates at different points in time. Specifically, here's the important part. Specifically, evolutionists have argued that mitochondrial DNA mutation rates have been slower in the distant past. That's the key. The rates have been slower in the distant past. An evolution which could, in theory, reconcile the erroneous predictions of the previous chapter with actual mitochondrial DNA differences. We talked about why Jeanson's calculations on those predictions are wrong and why the predictions are actually, the evolutionary predictions are actually correct. So I'm not going to rehash that, but that's what he's referencing. Yet for nuclear DNA, the slower rate in the distant past would aggravate the magnitude, blah, 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 blah. Does the molecular clock speed up or slow down? Can the evolutionary model predict when it accelerates and when it doesn't? Or will time dependency always be an idea that is retrofitted to any result as needed? A time dependency did it type of explanation. Well, there's two things I want to point out here. And the first is this right here. Specifically, evolutionists have argued that mitochondrial DNA mutation rates have been slower in the distant past. That is arguably the most egregious straw man of this entire book. It's difficult to find the words to describe just how absurd that is. That is not what anyone says. Nobody is looking at mitochondrial mutation rates and saying, no, the polymerases have actually gotten worse over time. That's what's happening. We're actually making more errors now than they were in the past. That's not the evolutionary explanation. The evolutionary explanation, the correct explanation, I will say, because none of this stuff should be controversial, it's obvious, is that you have a set of mutations that occur within an individual, within a generation. Not all those mutations are going to persist. There's lots of levels of filtering, right? So within an individual, right, I got a lot of cells, but most of them aren't going to pass on my DNA to my offspring. Only the germline cells have the potential to do that. So you have what's called the germline bottleneck. Most of the mutations that occur in my body could never be transmitted to my kids. So you have germline bottleneck. You also have selection, right? Some individuals are better suited than others. When things are selected out, they're not going to be passed on, and they could in fact be lost from that lineage. You also have genetic drift, the random loss of alleles from a population. Now you're cutting out more of those mutations. We're talking about nuclear DNA here. Inbreeding. Now you're going to lose more diversity via inbreeding. So you have all these levels of filtering out mutations that occur. So over longer periods of time, fewer mutations are going to persist within a lineage. And the long-term rate of accumulation, the rate at which mutations fix in a lineage, that's called the substitution rate. And that's the rate at which mutations occur and stick around and become stuck after you go through all those filtration steps that I just talked about, and some that I didn't talk about, right? I don't know if Jeanson doesn't know that. If he doesn't, it's completely unacceptable, inexcusable that he wouldn't know that. I don't know if he doesn't know that or if he is just misrepresenting it on purpose, but either way, it's completely unacceptable. This is just not even, this is just wrong. This is just wrong. It's bad. It's sloppy, or it's dishonest, and I don't know which it is. But you shouldn't take it seriously. The next thing right here, the second part of this is down here. Can the evolutionists predict when the clocks are going to speed up and slow down? Now, when we're talking about substitution rates, which is the correct thing, we're talking about accumulation in a lineage. Turns out the answer is yes. We can make those predictions. I will give you an example. I mentioned this yesterday. It's some viruses that infect dogs. Canine parvovirus 2 and 2A, I think, are the two viruses that I'm thinking of. These are viruses that emerged in the late 70s and early 80s. So they first appeared in the late 70s and they spread around the world. And by like the mid 80s, they were everywhere, every continent. dogs on every continent had them. And what you expect in that situation is, as they're adapting to a new host, you would expect what's called adaptive evolution. You would expect selection to preserve new genotypes that are beneficial in the new host. And that's going to elevate the substitution rate. And then once they kind of reach a fitness peak in that new host, you expect selection to switch over from adaptive to what's called purifying selection, meaning you're selecting against new variants because you're at a fitness peak. So any deviation from that is going to make you worse, right? If you picture it like a curve, if you're at the top, any mutation makes you worse. That's after you've adapted to your new host. But when you start at the bottom and then you adapt to your new host, when you're going up, selection is preserving more of the new mutations that occur because some subset of them are going to be adaptive. So the rate at which mutations fix in that mid-adaptation population, the rate of substitutions will be higher than the already adapted population or the ancestral population in the old host. We can make specific predictions about that. Like, that's a thing you can do. You could watch this happen during viral pandemics when a virus adapts to a new host. I'm going to make a prediction. The rate of mutations that we're going to find happened in SARS coronavirus 2 was faster during this pandemic than the substitution rate of that ancestral virus in the ancestral host species. Clear evolutionary prediction. I bet that's going to be the case once this is all cleared up and we actually really work on, get a good view of what these genomes look like. I bet we're going to see that it adapted within humans and therefore experienced a higher substitution rate during the first 6 months to year of this pandemic. Date and time, write it down, evolutionary prediction. I don't care if we didn't test it yet. You all have to take me seriously because I'm right until proven otherwise. That's how science works, according to Nathaniel Jeanson. You all see how stupid that sounds? That's the logic that Jeanson uses. When you apply it to actual scientific questions, you sound like a lunatic. But that's the logic he's applying for creationism. So, yeah. Okay, we're still in chapter 8 here. I promise we're going to finish this tonight, everybody. I promise. Okay, actually, evolutionists modified this narrative more. If we put the human-chimpanzee split at 4.5 million years ago, a constant rate of mutation would produce 6 million DNA differences. Evolutionists bring this number down to 4.31 million by invoking changing population sizes. So what we're doing here is that Jeanson is basically saying it's unreasonable to say the population changed, and that's just an ad hoc kind of thing. The thing is, we can figure out population sizes based on genetics. We can look at fossils, we can look at genetic diversity, and we can see, okay, how long has this thing existed? What's the mutation rate? What's the substitution rate? How much diversity do we have? Therefore, did we have bottlenecks in the past? Did we have a big population or did we have a small population? When you do phylogenetics, one of the parameters you have to set for your model is the population growth. Is the population going to be constant? Is it going to be linear? Is it going to be exponential? You have to set the demographics for your population when you're doing your phylogenetics. We're not just guessing for no reason. We're looking at all the different parameters, and then we're seeing what ends up matching what we observe best. And when we do that, we find not a constant population like Jeanson seems to think that we need to invoke, but a variable population size over time. It's not hard. Maybe he doesn't know, maybe he doesn't care. I don't know, but it's not hard. It's like, learn this stuff, Jeanson. Okay, down here at the bottom. But evolutionists can invoke pre-existing differences. Why can't creationists? He's talking now about created heterozygosity. If evolutionists explain 88% of our differences as pre-existing, is it too much of a stretch to bump that up to 99.6%? So this is where we're getting into some really fun stuff here. I just did not that long ago a video on created heterozygosity. I actually talk a lot about Jeanson in that video because what he does is a lot of hand waving here. What he's saying is, well, you've got all this variation in humans, but you say, well, 88% of that's inherited from our common ancestor with, chimps and other primates and yada, yada, yada. Creationists, we're doing the same thing. We just think it's due to the created diversity in Adam and Eve, and we think that created diversity explains 99.6% of the variation we see. It's kind of like that ice age thing that I talked about in part 1. Hey, you say there are ice ages, we say there are ice ages. We say there's one, you say there are a lot. Tomato, tomato. Same thing here. He's just asserting that his assertion, 99.6% due to created heterozygosity, should be on equal footing with the actual science-based number of approximately 88%. We have observed the human and chimpanzee nuclear DNA predictions. Evolutionary divergence time does not predict mutation rates. The human-chimp divergence time was revised in light of the mutation rates in these species. Furthermore, he's still misusing mutation rates, by the way. He means substitution rates. Furthermore, evolutionists explain the vast majority of human-human nuclear DNA differences, not by mutation, but by pre-existing differences. With respect to other species, mutation rates and divergence times run into additional problems. So there's a couple problems here. One is that the rate is not assumed. He's saying that we're assuming a mutation rate and then backfitting other data into that rate, but that's wrong. The rate he's talking about is substitution rates. That's directly measurable. You can do that because if you have two populations, you know how long in the past they diverged. You could do that with humans based on like archaeology and recorded human history. Then you could figure out the substitution rate and then apply that more broadly. Okay, that rate, say you do one, like you do this for island populations, like Vanuatu was settled, I think like 3-ish thousand years ago. So you can look at Vanuatu versus I forget what population is the sister group to the Polynesian Islanders of Vanuatu. You can look at the differences between them and then say, okay, that accumulated in the 3,000 years since they've been apart. What's that rate? Now, if you were to apply that over 50,000 years or 100,000 years, the rate's going to be slower over those longer periods of time, because of those filters that I talked about, things like selection and drift. But the rate that you observe, that sets your ceiling. And you know that the actual long-term rate will not be higher than that. It could be lower, but it's not going to be higher. Jeanson just ignores all this and says we're just asserting it because it has to work with our math. Like he's just getting it wrong. The second thing is that we're not just basing the mutation rate in a vacuum. Even if you put aside what I just said, there are fossils, and we can date those fossils, and we're really good at that. That part's pretty easy. Once you can date the fossils, that kind of sets bounds for your divergence times, right? So Jeanson's just making it seem like evolutionists are kind of making up numbers to fit the story they want to tell. But what he's actually doing is ignoring a bunch of evidence. That's the long and short of it. All right. If nothing else, it demonstrates that evolutionary divergence times do not consistently predict mutation rates. Again, he means substitution rates. And yes, that is the case because there's things like selection and drift and inbreeding. So along a branch on a phylogeny, depending on what's going on that branch, if there's strong selection or weak selection, then you're going to have different levels of mutation accumulation. Okay. You can even have two branches where one population is under purifying selection, the opposite lineage is under adaptive selection, so you have faster substitution rates in that second population. The divergence time is the same, but it's asymmetrical in terms of where the mutations accumulate due to the different dynamics affecting each of those lineages. Jeanson just wants to make everything consistent for, no, I don't know why, but he's wrong to do so. Okay, he's still talking about created heterozygosity here, and we're going to get into some stuff that's really fun. So under the model of pre-existing nuclear DNA differences, these bottlenecks would have been population bottlenecks, not genetic bottlenecks. Population bottlenecks, not genetic bottlenecks. Think about that for a second because it is a contradiction. In other words, population bottlenecks tend to eliminate large amounts of genetic variety only when the population size is kept small for many generations. A single generation population bottleneck followed by exponential population growth, e.g. the kind of event that describes the bottlenecks at creation, the flood, and the Tower of Babel, would result in the loss of far fewer varieties. What he's saying here is that when you have a big population and it gets really small, right? So you go from a lot of individuals to a handful of individuals. As long as it gets really big, really fast again, you don't lose that much genetic variation. That is bullcrap. The relevant thing through a population bottleneck is the effective population size. It's in, like when you do population genetics, it's N sub E. It's effective population size. That's the relevant measure through a population bottleneck. Now, if you have a bottleneck that is a small effective population size for 100 generations, compared to one that is the same effective population for one generation, then yes, you will lose more diversity in the longer bottleneck. But the effective population size sets a ceiling for how much diversity can get through that bottleneck. So, like for the flood, for example, you have Noah and his wife. That's a maximum for each locus in their genome, one or two alleles per person. So you've got 4 possible variants for Noah and his wife. And then you have the son's 3 wives, 2 each, if they're heterozygous maximally. So that sets the maximum at 10 alleles in that population. Now you can say, well, within each of those genes, you have lots of variation, and then they recombine and you can get more combinations. We're going to talk about that in just a second, because that's the exact case that Jeanson makes. And it's wrong. So this is now talking about Y chromosome and mitochondrial and nuclear DNA. So we kind of have 3 categories here. And he says, in both compartments, the nucleus and the mitochondria, all modern DNA differences are the result of mutations to the sequence that was present in the first ancestors. Both compartments go back to a single ancestral sequence, the mitochondrial DNA sequence and the Y chromosome sequence. And I want to just highlight a line on the next one, because this is something else right here. Thus, both the mitochondrial DNA and the Y chromosome sequences have the potential to act as strict, absolute molecular clocks. What he's implying there is the rest of the genome cannot act as a strict, absolute molecular clock. That would mean we cannot do coalescence analysis for any other part of the genome, because it has to act as a strict molecular clock in order to be able to do that. In order to be able to take, okay, we have these 2 things, how far back in time would they converge given the rate of change? What he's saying right there is you can only do that for the mitochondrial DNA and the Y chromosome, but not for the rest of the genome. Maybe he doesn't know that we've done those calculations for lots of things. Maybe Nathaniel Jeanson, who has a PhD from Harvard and talks about this stuff for a living, doesn't know that you can do coalescence analysis, which requires molecular clock calculations for lots of different parts of the genome. I suppose that's possible. Draw your own conclusions. I will just say that we've done this for the X chromosome, for example. It's about 500,000 years for the X chromosome. It's over 1 million years for the hemoglobin gene. You just keep going through them and giving you more dates, because we've done it for a lot of things. All right. Here we go. Thus okay, same thing. Same point I just made. He repeats himself. Moving on. Okay. However, in a more qualitative sense, the results that have been obtained thus far for the YEC model suggest the majority, if not the vast majority, of differences were pre-existing. So now we're back to created heterozygosity. And in a minute, we're going to get into how Jeanson does his calculations. That's what he does in chapter 9. I'm going to tell you why he's lying to you about that. He's just pulling a bait and switch, and we're going to get there. So let's keep going. To clarify, the prediction, this is my favorite line in the whole book. This is my favorite line in the whole book. If nothing else, just whatever, we're 38 and a half minutes in, mark it right here. This is the best line in the whole book. To clarify, the predictions I made above for animals naturally has an exception for humans. Naturally has an exception for humans. Creationists have long explained human origins apart from primate ancestry. So all the stuff he's talking about diversity, about common ancestry within families, about rates of change, about how you can look at created heterozygosity and the original mins, the original arc kinds, how that leads to all these different species. He's just going to say, but hold the phone, this doesn't count for humans, because humans are different, because creationists carve out an exception for humans. Why? Because we said so. Do we have a reason for doing this? No. If you read through this book, he talks about how these techniques work. He talks about things like you could compare humans and chimps. Earlier, I showed you an example talking about pre-existing differences. talks about them the same way he talks about nested hierarchies for other groups. But right here, this line, this invalidates his entire thing, and this is where the mask slips. And this is something that happens with Jeanson in his writing. It happens at the end of his 2015 AIG blog post, the whole genome mutation rates confirmed, D-loop rates or whatever. He says at the end, oh, we don't know, maybe they're somatic, but we're going to go with germline. That was the mask slipping. Right here, gives up the game. He says the quiet part loud. Humans don't count. Don't worry, humans don't count. Okay, humans, it doesn't apply to humans, right? Just like you can't just carve out an exception just because. Like that's not how science works. Either it works or it doesn't. You can't just say it doesn't count for humans. And the fact right here that he's just straight up saying, by the way, this doesn't count for humans because creationism. Okay, the fact that he's saying that shows you that this is all about presupposition. It's not about the evidence. He gives it up right here. This is the most important line of the whole book. So who's having fun? I'm having fun. All right. So still in chapter 8 here. We're just going to look at this, looking at pseudogene function in roundworms, humans, and fruit flies. Pseudogenes manifest the same evidence, i.e. they have transcription and they get trafficked to other parts of the genome. In fact, in fruit flies, 89% of the entire genome has preliminary evidence for function. We're back to encode now, by the way. We're talking about how other things have the same evidence for lots of function. In other words, experimental results are accumulating trajectory for pervasive genome-wide function. Given this trajectory, I would argue that the YEC explanation for DNA differences, that the majority were pre-existing and functional, looks promising. I will say that this is another thing this book does, or I should say Jeanson does in this book, is that he jumps around. So we already talked about encode. We already talked about genome function. Well, he's bringing it back here. It's not really related to this topic of created heterozygosity, but it's still in there. So yeah. All right. Now we're up to chapter 9. Finally, we're up to chapter 9. This is a short one. There's only a few things I want to say about chapter 9, and then we'll get to chapter 10, and some of those are pictures, and they're fun. So here we go. Furthermore, since these numbers represent the situation for so we're talking about created heterozygosity, and we're pretending to do math is what's going on here. He's saying, since these numbers represent the situation for a single lineage, and multiple lineages have descended from Adam and Eve, massive number of versions of each chromosome could exist today. Basically says, look, if Adam has 2 different versions of each chromosome and Eve has 2 different versions, and they recombine and they mix and match, well, you get lots of variation in the lineages that are descended from them. Well, he doesn't provide any numbers showing like that you could actually generate the allelic diversity, and I'll show you why he doesn't do that. It's because he does a bait and switch here. So here's what he does. In other words, calculations on the theoretical combinatorial diversity from a given set of chromosomes are best done using formulas for combinations rather than permutations. This is important because what he's doing is a classic bait and switch here. Because what the creationists need to be able to explain is, how do you get 1,000 different alleles for example, the ABO blood group? In my video on created heterozygosity, that's the gene I talked about. Creationists like to talk about the immune system genes in this context, but the mechanism for that's actually different. But you look at the ABO blood group, there are over 1,000 variants. Most of them are very rare. It's a single nucleotide polymorphism within one of those 3 main alleles, A, B, and O. And how do you get that from 4 initial alleles? Even if all of those differences are present in the initial alleles, in order to get all of those variants, you need to have recombination within the gene, and specifically within the exons of the gene. Now, intragenic recombination is rarer than recombination as a whole. Most recombination events don't happen within genes along a chromosome. Intraexonic recombination within exons, is even rarer. And the rate of recombination per chromosome per generation, per meiosis, I should say, per going from your gametangia into your gametes, that you go from diploid to haploid, that's when recombination occurs, that 2-step cell division called meiosis. And the rate of recombination in that process in humans is approximately 1.2 recombination events per chromosome. Now, genes are tiny compared to the chromosomes they're on. If you need to get 1,000 different variants within a chromosome from 4 initial alleles, you need to have millions of recombination events up and down that chromosome. And they're just, given the rate and human generation times, there just isn't enough time to generate all that diversity. Jeanson addresses this not at all. He pretends that problem doesn't exist by saying it's about combinations rather than permutations. What he means, because he switched language, he switched from bio language to math language. And what he means is it's really easy to generate lots of different combinations of alleles in different genes. In gene A, you get alleles, A1, A2, A3, A4, whatever. Gene B, B1, B2, B3, B4, but whatever. It's easy to get A1 with B1 and B2 and B3 and B4, and all those different combinations. Sure, yeah, you can do that. That's pretty straightforward. The problem is all the variants of A, starting with a 1, 2, 3, 4. How do you get A 1,000? Right? You need to do recombination within A, and there just aren't enough generations, and there aren't enough humans packed into the Bible story in order to make that work. And it's not close. It's not like, oh, you need an extra 10,000 years. No, you need millions more years. Okay, not that many. You need, hundreds of thousands of years. I think it's about 500,000 years is the calculation. This is something called the time to most recent 4 alleles calculations that Dr. Joshua Swaminas has done. I can actually link that in the description afterwards so everyone can take a look at that. Those calculations are taking into account things like recombination rates and recombination hotspots and that stuff, which Jeanson just totally ignores. So that's what he's doing here. It's a bait and switch, pretending to be able to address a problem that he's actually completely ignoring. Okay. So I just like this line because it's so oversimplified. In other words, given a set of heterozygous ancestors, a fantastic potential for chromosome diversity exists. Since chromosomes encode visible traits, these results imply that heterozygous ancestors also contain a fantastic potential for genetic diversity. This is the kind of grandiose generalization that is going to appeal to a non-scientific audience, which again is why I think this book is targeted at creationists who are not scientists. It's to reassure them, and this is the kind of passage that's going to do that. Trust me, I know I'm a scientist, and chromosomes code for traits, and if you have lots of variation, you can just mix and match and get lots of variations of those chromosomes. Therefore, you can get lots of traits. Problem solved. The thing is, I don't care about phenotypic diversity. I'm not complaining about that. What I'm complaining about with this math is allelic diversity. How many different alleles can you get from an original set of 4 in the amount of time you have to do it? And the answer is not as many as we have. That's the answer. All right. If human breeders can do so much with so little genetic potential, now we're getting into this idea of genetic potential, and this is fun because he talks about speciation, but he's wrong about it. If human breeders can do so much with so little genetic potential, and if much more genetic potential exists in the wild, then natural substitute for humans is almost unnecessary. Natural processes need to accomplish so little. The relevant phrase here is genetic potential. He's now getting into this idea for speciation that you have a pre-existing kind of pool of genetic potential at the start, and then as you speciate, you lose it. And this is going to get into what he talks about with like speciation rates. It's kind of an out there idea, and we'll get into it. Okay. In short, under the model that I just described, formation of new species is easy as long as subpopulations historically broke away from the original ancestral populations, as long as the breakaway populations move towards homozygosity, as some of the original heterozygous DNA loci, such that isolated distinct populations appear, then new species would have formed. The interesting thing here is he is assuming that speciation events must involve a move from heterozygosity to homozygosity. In other words, speciation events must involve a loss of allelic diversity within a population. That's not the case. You can have things, for example, like adaptive radiations, where you have unoccupied niches. You have resources that are available but aren't being used. Well, when a new species diversifies to occupy those niches, that often involves new alleles that allow that species to exploit those specific resources that the ancestral population was not exploiting. If you're introducing new alleles, you are not moving from heterozygosity to homozygosity. You're increasing the allelic diversity, not decreasing it. So I don't know what he's getting at here. The second thing with that is, even if the process I described, even if those new alleles go to fixation, they're not pre-existing in the population, right? The new alleles that happen via mutation that allow you to exploit a new resource, those were not pre-existing. So you haven't lost something that existed in the past. So Jeanson's just, I don't know what he's describing here, but it's not how speciation works. Or at least it's not always how speciation works. Certainly it can happen, but not genome-wide typically, and not in many cases. It depends on the ecological conditions, which is a problem that we're going to see again, him ignoring the ecological conditions. So, no need to sequentially isolate or select individual DNA variants from generation to generation. Yeah. Evolution is a parallel process. But you know what he's not doing here? He's not showing his math of how his model works. He's just saying, oh, you know, it's fine. You don't need to like do this. You just have lots of lineages and they're all doing stuff at the same time. You don't have to do it one and the other. Like, okay, yeah, that's true. It's a parallel process. It's not a serial process. Great. Show your math. He doesn't. Somebody last night in the chat asked if there are like equations in this book. And the answer is no. And I forget who it was. I think it was CRISPR-Cas9, actually, if everyone knows CRISPR. And I said, if you don't have equations, you're not doing population genetics. And that's what's going on here. He's gesturing towards population genetics, but he's not doing anything that's actual population genetics here. Okay, now we're into chapter 10. This is the final chapter, everybody. So we have 19 more. So we got a bunch, but some of them are pictures, and the pictures are funny. So yeah, it's taking time, but that's okay. We'll finish. Okay, so as per chapter 7, the fact that a clock might appear to be a premature conclusion. The fact of a clock might appear to be a premature conclusion. Recall that neither side has solved the discrepancy between their predictions and actual mitochondrial differences among vertebrate species. See that false equivalence again? He's setting up a straw man for what evolutionary predictions should be, saying, oh, look, those predictions are wrong. Looks like neither one of us has an answer. Who's to say? That's what's going on right here. All right. Nevertheless, both sides in the debate would concede that a clock exists. Yes. What the correct side says, what the science side says, is clocks exist, and we know how fast they operate. And in fact, we can compare different lineages and different types of organisms and see this. We know what the rates are. We know what the rates are. It's like we've measured this stuff, and he doesn't want to admit it because the numbers show that he's wrong. But that's just the truth of it. Creationists and evolutionists agree that branch points, phylogenetically, when you do phylogenetics and you get branch points, approximate speciation events. They must, since mitochondrial DNA trees are used in part to define species. Thus, for vertebrate species within a family, both creationists and evolutionists agree that mitochondrial DNA trees represent the history of speciation within the family. But what about outside of the family? Because we can do that same math for phyla, for kingdoms, for domains. Stuff in the mitochondria, for example, is really well preserved. So, like, why can't we do it outside of families, Dr. Jeanson? Why does it just arbitrarily stop there? I think I know why, but I don't think there's a good biological reason for it. Where are we? Here we are. Okay. But, oh, see, here we are. But where do we start where do we put the start of the speciation process? Since we're limiting our focus to living species within vertebrate families, two things here I want to point out, and it's this word: living species, living within vertebrate families. So again, we have the families, arbitrary distinction for no reason, and living. There's a lot more diversity in extant families than is alive right now. There's a lot of extinct members of a lot of families, and that's going to come into play in a few minutes when we talk about speciation rates. Yeah, it's just the insistence on living is weird, given the argument that he's making here, that we can predict speciation rates from the arc to the present based on the living diversity. There's a lot of extinct things that we have to consider when we're looking at speciation rates. That is a small part of Jeanson's problem here, but it is by no means the only problem. Okay. So how can we be sure we're picking the actual start of the speciation process? In short, we can't until we measure the mitochondrial DNA mutation rates in all of the species we're examining. However, we can make a simplifying assumption and then conduct a preliminary analysis. You know, this is going to be good, right? I've already made this simplifying assumption on the equid mtDNA tree. The assumption invokes a technique called rooting on the midpoint. This method takes the two most divergent branches and sets the root of the tree as halfway between the two tips. Effectively, this strategy helps minimize variety in branch lengths, and it seems to have done so for the equid tree. Now, this is a little bit technical. But what he's saying is phylogenetically, we're making assumptions about where the common ancestor would be in terms of its sequence. He's saying you take the two most divergent things, right, and you put the common ancestor between them. Kind of the consensus of the kind of the average of those two. I'm oversimplifying, but that's basically what this means. There's a lot of assumptions built into the technique. Sometimes they're valid and sometimes they are not. So, for example, I've mentioned earlier things like adaptive radiations, or just a simple example of a species adapting to a new niche. Well, you would expect, and in fact, we observe faster mutation accumulation in those lineages than lineages that are already well adapted to their ecological niche. So, like, for example, you have, let's take chimps and humans, for example, right? So we know that the common ancestor of chimps and humans, that would be something that was a tree dweller on all fours, right? It wasn't an upright walking, biped kind of thing. So humans, as we moved from forest to grassland and we started walking upright, we have a lot of things that accumulated. What we might predict based on the fact that chimps basically stayed in the same environment and humans moved into a new environment, a new ecological niche, you might predict more mutations accumulating, a faster substitution rate in humans as we adapted to that new, or I should say in the human lineage, as we adapted to that new environment. Now, we don't have the genome of that common ancestor to check, so we can't test that prediction. But that's the kind of thing you would assume, and you wouldn't root at the midpoint in that situation. That would get you the wrong answer, right? So he's saying we're going to make this generalized assumption when it's completely inappropriate to do so. Even so, his numbers, as we'll see in a moment, are hilariously wrong, as we'll see. But that's what's going on here. So here's what he's saying. Now we're going to get into this because we're going to see how he backs up this claim, because this is one of the only places in the book where he backs up a claim with actual numbers, and it's so bad it's funny. So in fact, in each mammal family I examined, I found strong evidence of a linear pattern of speciation. Among the remaining mammal families with higher numbers of species and with mitochondrial genome representation of around 40% of these species, the same results ensued. And then he goes through a bunch of species and he says, you plot out the number of species over time, and you get a linear rate of speciation. So let's look at, I think the pictures are next. So let's look at some of these graphs and see if it looks linear to you. And a bit of a spoiler, I have put my yellow lines in there as I was annotating these as I was reading this book. So he kind of gives away the answer, but like, that's not linear. Come on, Jeanson. Come on, really? Like, that is clearly showing punctuated equilibrium. Okay, what we have here with a lot of new species, that's an adaptive radiation event where you have a bunch of diversification. Here, this is where you have kind of stasis in an environment and everyone's pretty well adapted and there aren't any available ecological niches. Like, yeah, you can draw a linear regression line there, but like, why? No. Here's another one. You can see there's basically a bimodal one where you have a higher rate here, and then it slows down right about there, right? And then you get a much lower rate there. But we're just going to like pretend that that's linear. Like, yeah, I can get R squared to 0.9. Like, fine. That's not a linear rate of speciation, all right? Here's one where it's stasis, rapid, stasis. There's 3 distinct periods here in the ecological history of this group. This is in the mouse and rat family. And there's like obviously 3 distinct ecological situations going on here where you have a period of slow speciation, you have a period of rapid speciation, and then you have another period of slow speciation. And we can just keep going. Like, are you kidding me? Obviously, this one is a much poorer fit for the linear one. Like, that's not even close. Come on. And then this is what I love here is can we appreciate just what he says here? Shows resemblance to a linear function. shows resemblance to a linear function. This is how I know that Jeanson's credentials are legit, because the only way you can write that is if you have experience trying to get results that are not statistically significant published. That's how you write a line like this, shows resemblance to linear function. Jeanson learned how to do that in grad school. How do I know that? Because I learned how to write like that in grad school too, because that's just how life is. I just love it that he's going to show this as part of his linear speciation thing. There are more graphs like this throughout the chapter. Now, in fairness, some of them are better. This is a lot better. That's much closer to linear than some of the other ones. This is the line I want to point out here. Because nearly all of these patterns were linear. Do I need to show the pictures again? I don't think I do. I don't think those are all linear. I think Jeanson's using a linear fit. I can draw a line. That makes it linear. No, those are not linear speciation rates. He's assuming they are, but they're not. Now let's keep going because there's more fun to be had here. Also, that's not linear. This is the equid family. What's going on here? It jumps up and then it slows down. This is like it's like kind of like that. But here, I want to just show this right here because what he's doing is he is looking beyond mammals. And here are the criteria for the groups he's looking at. I selected families which had more than 12 total species, fine. At least the same number of species represented in, you got to have enough data, fine. And 3, little branch length variety to minimize statistical uncertainty. I'm selecting groups that have little branch length variety. Now, branch length variety means that you're going to have different numbers of mutations accumulating in different lineages. What might that be a sign of? Anybody remember? It's a sign of having different selective pressures acting on different lineages within a group. One might be adapting to a new environment. The other might be well adapted to its environment and experiencing purifying selection. Well, under that situation, maybe you have in one lineage stasis and in another lineage, you have rapid speciation in a new environment. What Jeanson is doing here by setting these criteria, and again, a non-scientific audience is not going to catch this. What he is doing here is cherry picking to make his desired conclusion more likely to be correct. He's saying, I predict that, I don't know a good one. I predict that I'm going to pick crayons at random and they're mostly going to be green. And then the box of crayons he picks is the one is like the rainforest collection pack or like, I don't know. I don't have a good example of this, but like, that's the kind of thing he's doing. He's biasing his sample set to be more likely to get the results he wants by setting these criteria. That's what he's doing here. Yeah. It's just straight up, just cherry picking on the front end instead of the back end. So instead, nearly every family we examined, the pattern of speciation was a straight line. Now, first of all, I showed you those were not straight lines, and I didn't even show you all of them. But second, for the non-vertebrate ones, he's cherry picking. And if you examine the ones he excluded, you would get even fewer straight lines, right? All right. Now we're getting into some fun stuff because we're talking about some bonkers idea that has no basis in reality called speciation potential. The linear pattern implies a similar conclusion, but in the realm of speciation potential. Specifically, because the pattern of speciation is linear in each family, the rate of speciation per family is constant. This is the mathematical nature of linear function. Okay? Speciation potential is not a thing that can be quantified because it's not an inherent characteristic of a genome. There's not some fixed level of variation within a genome or a population that allows that population to form a fixed number of new species. That's not how this works. Framing it that way, framing it as you start with a bunch of potential and then you have speciation, and as you speciate, you lose that potential, that is begging the question. It is assuming a creationist framing. By assuming a pre-existing set of diversity that you cannot add to, and then a loss of diversity as you speciate and propagate following the radiation from that common ancestor. So that's the setup for this argument. The actual argument itself is not good because this makes no sense. He says, now consider again the lottery analogy and apply it to species. For example, in Bovidae, when speciation first got going, when did speciation start? Evolution is an ongoing process. Sometimes that involves grades of reproductive isolation and a slow decline in gene flow between populations. Does that count as speciation? Hard to say when it starts. He's implying that there's like speciation starts. It doesn't work like that. But let's put that aside. Only a few species are present, yet the linear function demands that 0.1 new species form per mutation. In other words, these few species would have formed new species after a certain period of time. So species have to form in a linear way, independent of the environmental conditions. With few initial species present, each one was virtually guaranteed to form another species. However, as time progressed, the number of species increased, but because the rate stays the same, you have more species, therefore, the chance that any one particular species formed another one got lower and lower. Just like the lottery, the more that participated in the speciation process, the less the chance that any one species would win the speciation lottery and form a new species. Like, what the hell? What is he talking about? This is just basically what he's saying is you start with some pre-existing capacity to speciate. And then over time, as you speciate, each subsequent species, each new lineage has a subset of that initial pre-existing capacity, and therefore the per lineage rate of speciation decreases. But because as they divide, you have more lineages, the overall rate of speciation that occurs stays the same. You get a constant linear rate of speciation. It's difficult to impress how much that doesn't make any sense. He's treating speciation as a mathematical function, not an ecologically dependent process. If you're in a constant environment and all the resources are being used, all the niches are occupied, you're not going to have much speciation. If you're in an environment with lots of available resources, lots of unoccupied ecological niches, you're going to have rapid speciation in that environment. It's ecologically dependent. It's not just a function of time. The fact that he presents it as a function of time independent of ecology, I don't know what to do with this. Is another example where I don't know what to do with this because it is so blatantly, obviously, and hilariously wrong. There's nothing else to say about it. There's nothing about this that bears any resemblance to the reality of how speciation works in nature. Like, it's bad. I don't know what else to tell you. This makes no sense. So we're basing, and just to connect this to the broader theme of what Jeanson's doing, he makes bat poop crazy assertions like this, and then builds his model on top of these assertions. And like, no, this is nuts. This just does not work. Like, just no. All right. So now let's jump down to because we're not done with this page. We're at the same page at the bottom. In other words, whatever caused species to form initially seems to have declined in its abilities with time. The potential for speciation seems to have been diluted. In short, the ancestors of modern species look like they had much more potential for speciation than any of their descendants do today. What he's trying to say is as he described earlier, you go from really diverse to really not diverse. Now, that's not true. We observe the opposite of that. It depends on the case. But what he's saying is you inevitably go from more diverse to less diverse, and therefore your ability to form new species decreases over time. Again, he's treating this, now he's connecting it to genetics in addition to time, but he's still leaving out the ecological aspect to it. You need to look at resource competition and what ecological niches are available within your environment. And furthermore, how these processes reinforce each other. Because if one species adapts to a new niche and it starts eating a new plant, for example, well, now that new herbivorous species, that itself becomes a new niche, and you can now have potentially a new carnivorous species that eats the new herbivore, right? So it builds up on itself. And this is, for example, what happens during adaptive radiations. Ecological complexity is a positive feedback loop. So like, for example, during the Cambrian explosion, which creationists love to harp on, that evolutionists can't explain. No, it's pretty easy to explain that. One, we know based on the living things that there was pre-existing genetic diversity before the Cambrian. Two, Environmental diversification. You go from a fairly constant environment to a highly variable environment. Now you have more resources. 3, things adapt to those new resources. 4, those things themselves become new niches that other organisms can exploit. This is where you get complex predator-prey relationships. It's a feedback loop of increasing ecological complexity. This is not that complicated. I mean, it's complicated if you like really get into it, but the big picture is like it's pretty simple to explain. An important component of that is the ecology. And Jeanson's just ignoring that. He's pretending that that doesn't have any role to play. We're almost done. We are almost done. We're only going to like 20 or 21, I think. So we got 6. We're on number 16 here. Okay. Sorry. Here we go. Okay. So to produce a new species, these traits and nuclear DNA combinations must be isolated. Yep, small population sizes, inbreeding, and migration can lead to the founding of new, more homogeneous populations. True, but that's not the only way you get new populations. As these more homogeneous groups grow to larger sizes, they might eventually be recognized as new species. True. Eventually, this process limits the ability of these new species to form additional species. Liar, liar, pants on fire. Okay, I don't know if he's liar, liar, pants on fire. He's wrong, wrong, pants on wrong. Pants too long. I don't know. Come up with a rhyme. Since under this model, new species tend to arise via shifts from more heterozygous to more homozygous states, these new species effectively lose a number of heterozygous variants. That's an assumption that's unfounded. That doesn't have to be the case. He's just saying this is the case, and it doesn't have to be. He's saying in my model, because reasons, this is the case, but he can't back that up. That's not what we actually observe. I should say it's not universally what we actually observe. If speciation potential resides in the ability to spawn genetically and visually diverse offspring, then high heterozygosity represents high potential. Low heterozygosity represents low potential. The process of speciation that we've derived naturally dilutes the speciation potential in descendants. If only there was a way you could get new variants within a population, if only that was possible. Is there a word for changes that occur in a genome, maybe due to errors that occur when copying that genome, or just spontaneous damage or damage from chemicals or UV radiation? I think there is. The word is mutations, right? So you get new variants introduced in populations all the time via mutations. And it's interesting to hear this argument from a young Earth creationist. I don't know what Jeanson thinks of something like genetic entropy, but what he's saying here contradicts genetic entropy. Because he's saying that there just aren't that many mutations. The ability to diverge is lost over time because there just aren't that many mutations. He says all variation, almost all variation, 99.6% of variation is due to that pre-existing heterozygosity. Okay, then we're not genetic entropying ourselves to death, according to Nathaniel Jeanson, I guess, because mutations apparently don't exist. Okay? That's what he's arguing here. And it makes no sense on its face, but 2, it's contradictory with a whole other set of creationist arguments, which I love when they contradict themselves. That's always endearing. All right, let's keep going. We've argued that shifts from heterozygosity to homozygosity are the major genetic mechanism of speciation. That is not true. That's just not, we don't have to spend more time on that. That's not generally the case. All right. I love this one. We're gonna talk about this one a bit because there's some pretending going on here. Okay. Let's see. He says, nevertheless, the zebra ass lineage split around 1/4 of the way along its evolutionary tree that he draws. On a timescale of 4,500 years, let's just assume this split would have occurred because you're dating from the arc 4,500 years. The split would have occurred around 3,400 years ago. Or around 1400 BC. We know from historical records that the ancient Romans were aware of the existence of zebras since the Roman Empire ruled only about 2,000 years ago, i.e. not 3,400 years ago. The speciation timescale is consistent with recorded history. This is quite the dance, because you may be shocked to learn that there were other Mediterranean civilizations prior to the Roman Empire that documented lots of biodiversity. For example, the Roman Republic, the Greek city-states, Phoenicia, Carthage, Egypt, a bunch of Middle Eastern empires, the Hittites, the Assyrians, the Babylonians. We have records from all these older civilizations of lots of biodiversity. He's saying, oh, look, you look at the Romans. Well, great. They don't have this biodiversity, the lots of different species existing too early in history to violate my speciation timeline. The problem for him is the Egyptians do. The Egyptians have tons and tons of species that exist within, if we're being generous, within 1,000 years of the flood, right? We have recorded Egyptian history, like hieroglyphics, even kind of pictographic writing systems that are before true hieroglyphics, going back to the Middle Kingdom and the Old Kingdom. But even if you forget the Old Kingdom, even if you just go with the Middle Kingdom, which is, I forget who it is in the Bible, but there's someone that's in the Middle Kingdom, might have been Abraham, I think. And in there, you describe a bunch of different types of cats, different types of raptors, right? Different types of wild, like sheep and goats and that kind of thing. Hippos are in there. There's tons of biodiversity. There's way too many species already at that point to be possible in Jeanson's timeline. And here's the thing. This goes back to what I mentioned before when he said living species within a family. You still have to account for the extinct ones too, because by the time you get to ancient Egypt and the Middle Kingdom, which, if we're being generous, gives you 1,000 years since the flood, if we're being super generous and ignoring the ice age that creationists say occurred in there, not only do you have to get all the extant members of Felidae, for example, you have to get all the extinct ones out of the way, because the extinct ones are not recorded in those records. So you have like 30-something living ones, and you have to get rid of the over 100 extinct ones also. They had to speciate and go extinct in that time frame. That's not possible using Jeanson's rate. It's just there's too many species in too little time. By putting it at Romans and only focusing on living species, he's ignoring that problem. But if you include all ancient civilizations and their earlier pre-Roman Empire records, and you also consider the extinct species, it just blows the timeline away. It's totally not possible. Jeanson's just pretending that's not the case. Continuing onwards, I think this might actually be the last one. Is this the last one? This is the last one, mercifully. There we go. This is the last one, everybody. The trajectory is upwards, implying that more species will form in the future. This is the last one because it actually relates to a specific prediction Jeanson has made. One, this is like the most mundane thing you could say. There's going to be more speciation in the future. Yeah, you think? Evolution is an ongoing process. So yes, there's going to be more speciation in the future. The other thing here is I want to just point out Jeanson's correct prediction. He has predicted his, whatever number of species per year or X number of years per species within a group. And he specifically did this for birds. And a few years back, there was a new species of finch identified, ironically, on the Galapagos Islands. And Jeanson said, look, there you go. There's my new species of finch. It's right in the time frame I expected. Knew we'd be discovering it soon. Have his process, you have divergence, you have loss of diversity, poof, get a new species of finch, just like Jeanson predicted. Here's the thing. That new species of finch, it's a hybrid. It's not the kind of thing that Jeanson predicted. It's not about, population got isolated, it became less diverse, and it became its own lineage, and now we consider a new species. That's not what happened. It's a hybrid finch. And now it's doing its own thing, and it doesn't reproduce with either parent species. It's not the process Jeanson described. So that process is separate from the one he described, so it's independent of the rate he proposed. So that example doesn't fulfill Jeanson's prediction. It just doesn't. It's just the wrong thing. It's a totally different process, and Jeanson just pretends that's not the case. And that's the last one I'm going to mention, because that's what I got. So I know this has been going, goodness, we've been going for almost 80 minutes again. Thank you all for watching. I apologize this has been so long. So let me just wrap this up with a quick summary here, so we can get rid of this. We don't need that anymore. There we go. We don't need that anymore. Let me just wrap this up with a quick summary of the problems with how Jeanson constructs his arguments here. Okay, so they work like this. There's three big things in part one at the beginning that I identified as the major problems. One, he bases his arguments on assumptions, oversimplifications, logical leaps that make no sense. Just basically, he's basing his arguments on things that are wrong. Some examples that I showed you: linear speciation rates. We're just going to assume linear speciation rates. We should assume constant mutation accumulation rates. He says they're mutations, but he's actually using substitution rates. And as I described, he's using the wrong units for how to calculate those. The genetics of speciation that we just talked about bears no resemblance to how that actually works in nature. What he describes is closer to the math of radioactive decay than speciation, right? He's not describing how speciation actually works. The second thing that he does wrong is just a rampant, egregious straw manning. In part one, I showed you his argument, what evolutionists say about bad design and how you can conclude that bad design implies evolution, and he just mischaracterizes the argument that evolutionary biologists make. He just doesn't represent the argument correctly. Another example that I showed you tonight was what time dependency means. It doesn't mean that the mutation rate slows down. It means that mutations are filtered out over time through a number of well-documented processes. And like, that shouldn't be controversial. Germline bottleneck, selection, inbreeding drift, those are all things that happen, and nobody should be surprised by that. And then the third thing that he does is either misunderstanding or misrepresenting how science works in a way that biases in favor of his conclusions and his model. So basically, the trick that he does is he makes a prediction, a testable prediction, and says, whoa, look at me, I made a prediction. You haven't disproven it. Got to take my model seriously. Can't tell the difference between my model and your model because I made a prediction and we don't know if it's wrong. That is the beginning of the process, not the end of the process. You have to test the predictions. And as I said, you hear this if you're involved with talking to and with creationists and reading and listening to what creationists say. You hear over and over again that making predictions, making testable predictions is the gold standard of science. That's the gold standard, making testable predictions. And Jeanson makes testable predictions. No, that's wrong. The gold standard is making testable predictions and then showing that they're correct. Jeanson doesn't do that. He just stops at making the predictions and doesn't make an effort to actually show that they're correct. So the takeaway from all of these problems with this book, putting aside like really digging into the biological arguments like EvoGrad did. And again, I want to plug EvoGrad series. It's linked down below. I also have a few videos that are more detailed in terms of Jeanson stuff linked down below. Putting aside like the nuts and bolts of why his like arguments are wrong, the way he structures them indicates that he's not trying to convince a scientific audience. What he's trying to do is reassure people that are already on his side. That's the impression I get as a scientist from reading this book. I don't feel like this book was written to try to convince me. It doesn't seem like he's making an effort to do that. What he's doing is trying to convince non-scientists that the thing they already believe has a basis in science. And he does that using a lot of hand-waving, a lot of, I think, misrepresentations, or maybe he just doesn't know things about evolution. And it comes off as just a really unserious bundle of arguments and, frankly, bundle of contradictions in some parts. So that's what I have to say about Nathaniel Jeanson's I think it was 2017 book, Replacing Darwin, I think it's called The New Origin of Species. Let me make sure I get the full title right. Replacing Darwin, The New Origin of Species. That's what I have to say about Nathaniel Jeanson's Replacing Darwin, The New Origin of Species. It's not good, everybody. It's not good. So let me the last thing I'll say here is that I've now spent, about 3 hours talking about this book. God bless anybody who's listened to this whole thing. If anybody has a problem with anything I have said here, as is always the case with anything I do, come on and tell me what I'm wrong about. I'll give you as much time as you want. That's what I will do. But you have to reach out and you have to like actually make the effort to try to correct me. So that's what I invite all of my lovely critics. If you want to show that I'm wrong, come on, tell me about it. And I do, especially if Nathaniel Jeanson happens to see this. Don't ignore my messages. I already sent you a message. get back to me, and we can set up a thing, because I would love to have Dr. Jeanson on and we could talk about these things, because you better believe I have questions. And with that, I will say goodnight, everybody. Thank you all for watching. I hope you all found that educational, and somehow maybe some of you found that even a little bit enjoyable. Thank you, everybody. Have a great night. And now let me awkwardly click over to the end broadcast button. There it is. Bye, everybody.